Ladies and gentlemen, our next speaker is Dr. Arup Ghosh. I'm sure all of you are familiar of him. Dr. Arup Ghosh is a senior principal scientist at the CSIR-Central Salt and Marine Chemicals Research Institute in Bhavnagar and is also a professor at the Academy of Science and Innovative Research in Ghaziabad. He is a renowned scientist, one of the authorities we believe in seaweed. And sir, I hand it over to you. Please do it just. Yeah, it's already there.
Thank you very much, Bipinji. I thank you very much for giving me an opportunity to speak in front of all of you, the august audience. Basically, I am from CSMCRI. It's a research institute. This is a CSIR Research Institute in Bhavnagar, Gujarat. And we work in many things that are related to seawater. So anything that is related to seawater that revolves around it. My specific topic for today would be seaweed biostimulants, brief about what are the bioactives, what are the mechanism of action, their effect on crop growth, quality, and something about the regulations also. So to start with, let me give a brief overview of what CSMCRI is about. So we work on marine products and processes, marine biology, marine environment. In marine products, we deal with salts, we deal with water, marine chemicals, both inorganic and organic, marine elements, and marine biology, we do seaweeds, microalgae, halophytes, plants which are only growing in high salinity condition, salt tolerant plants, and we try to develop even genetically modified plants that have salt tolerance property, marine microbes, marine bioactives, and different products, valorization of seaweeds and the microbial products. We also have experts who have great expertise in assessing marine pollution, EIA studies, and impact assessment studies. So that is in a nutshell what we do. And you can see that we are a CSIR institute, and technology is our main focus area. So technology we develop, and we have been, considerably doing good on different things like composite of patents, publication, societal work, and visibility. So we are in fact ahead to some of the very reputed institutes on these counts. This is a bit older data, but the recent data is also the same. And since I told that we are working with the industry, so we work with several different industries. We try to take the problems and then sort it out by different groups of scientists. We have about 90 scientists put together, apart from technical and other stuff. So that was in nutshell what we do at CSMCRI. Now I would be switching over to seaweed, biostimulants, and seaweed cultivation. You can see this is how seaweeds are cultivated. The productivity is much higher than any of the terrestrial plants per square meter area or whatever. The beauty is that it does not require arable lands, it does not require fertilizer, it does not require pesticides. So we as an institute have developed different technologies for cultivation, different types of seeds. You can see some are green, brown, and red seaweed, which is a basic classification about. And we try to develop commercially viable value added products like agar, agarose, alginate, carrageenan, and several other innovations out of these materials from seaweeds. So this is cultivation that is getting in Tamil Nadu coast, and this cultivation has actually helped a lot to the farming community, the coastal fishermen, pattern of usage of income has increased, the lives of the woman that has in fact changed, and in fact the numbers that accrue that are very important. Okay, so not all the methods are same for seaweed cultivation. It will depend on different coasts of India, because India has a huge coast. Not all the cultivation methodologies will be same. So this one is the earlier one I showed, raft method, that is suitable for Tamil Nadu. This one is suitable for places where there is high tidal current. So this is called tube net method, and that is how this seaweed, this is Gracilaria dura and Kappa ficus, that has been cultivated in Gujarat. A very recent development is we have been executing a project wherein pan India mapping of what are the conducive areas for seaweed cultivation has been initiated, and I think in another 6 months or so we'll be having an entire map of India where seaweed cultivation is possible, and that will be actually even and there'll be this report, that is a policy, seaweed policy, that is in the offing, and that will be very soon maybe released by our Niti Aayog. Coming to the technology, this the latest technology what CSMCRI has developed is a animal feed additive that is good for lactating cows, growing calves, egg layers, and broilers. We have done this in collaboration with different ICR institutes like Central Avian Research Institutes and National Dairy Research Institute, and we have toxicity trials also done. A good thing about it can also reduce methane emissions from the ruminant animals, improves the body weight gain of the growing calves, and definitely improves immunity and the productivity like egg production and meat production of chicken. Coming to the biostimulants, world over there are only few species which are utilized for making biostimulants. You can see Ascophyllum nodosum, Eclonia maxima, Durvilia species. These are most of the important ones. Even 90% of the biostimulant market which we have in India is totally based on Ascophyllum. But in India also we have different species which are growing, which can be grown, like Kappa ficus, Gracilaria, Sargassum, Turbinaria, and we are basically working on the Indian seaweeds, both their valorization, characterization. One thing is important, that composition vary with different types of seaweeds. So this is something very important from regulation point of view. You can see I am comparing only two of them, like one is your red seaweed and another is a brown seaweed, both found in Indian coast, Kappa ficus and Sargassum. And we see that there are several things like total flavonoids, ascorbic acid, phenols, antioxidant, different species of polyphenolic compounds that are also very different. But that does not mean that there is a change in efficacy, because there are some things which are not found in one sort of seaweed, and others may have some other bioactives. Even we have done a comparison also between the Ascophyllum and the native ones, and we found that it's comparable, and in some cases it is much better. So regarding the efficacy trials, that is also part of a regulation. We have actually carried out 175 trials. The requirement is only 3 trials, but we had carried out 175 trials. For example, in rice only, you can see we have carried out more than 20-30 trials all over the country by collaborating with different agriculture universities and ICR institutes. And with what Dr. Sanjay Kumar just now told, that it has to have a very specific statistical design. There are doses, like 3 doses. In these trials, we have done at least 4 doses for each of the seaweed biostimulant. There has to be proper replication, there has to be proper design, and then you find that we have found about 11 to 37% improvement in yield over and above the recommended doses of practices. We have done for maize, green gram, black gram. Again, the problem for regulation is for crop labeling, we have to have trials for each of the crops. But we saw that in EU there is a provision that on 9 crops, if you do, it is applicable for all the crops. That is, I mean, in Europe. So some sort of thing that will be definitely required. It has to be discussed with the regulators that how this can be done. For us, we did like 2 for cereals, 2 for pulses, 2 for oil seeds, 2 for sugar crops, like that. But I mean, the clarity is not there on this front. Not only on Carpa ficus, we have also done for Gracilaria. That is another seaweed that is found in Indian coasts. And same number of 175 trials for agronomic trials and another 165 to 70 trials. These are big scale demonstration trials on farmers' field. Similarly for sargassum, that is also we have done. This is not only good for plant, but it is also good for microalgae. Now this is the regulations, like what are the heavy metal contents. Even they have put the micronutrient contents, which are actually not very harmful. I mean, they are beneficial, but still they have put the limits, which is again different from EU regulations, because you can have that micronutrient included as a different and there are methods how you can do it, which here we cannot do. So these are the limits, and we can see that. See, I mean, now I'm just taking one example. We have done for many of the seaweed extracts. This is for only one extract. You see that there are a lot of constituents, including metals, heavy metals. For example, none of the heavy metals present in the seaweed sap will exceed the one that has been recommended by FCO. That we can always do. There are so many other compounds, organic compounds, bioactive compounds. You can see in the chart, belonging to several categories like sugars, sugar alcohols, amino acids, lipids, organic acids, amine derivatives, nitrogen compound. But do you want to test all of them? If you give this list to the regulators, are these bioactive compounds? For that also we have seen. See, these are the bioactive compounds that is found in Carpa ficus, but none of the regulators will have any method also to a standard method to actually evaluate them or even detect them. But are they bioactive at the physiological level? Yes, they have. I mean, even if you add one compound, synthetic compound exogenously that is found in literature, we have found that our seaweed biostimulants contain a lot many concoction of compounds which are in mixture. So they are not one, they are not two, there are many of them that is present together, which even exogenously, if it is applied, there are available literature that it gives effect. Okay, at the applied concentration I'm talking about, not the ingredient. But again, having said so, can we give? No. So there has to be some. These are the bioactive compounds. We cannot give them. So marker compounds, seaweeds will have markers. Polysaccharides are the markers. Okay, so these are some of the suggestions which we have time and again given. Okay, like carrageenan, look for carrageenan, look for alginate, look for fucoidans. So these are some things which we have discussed. I mean, it depends again on the regulators what they are going to do. But yeah, definitely there should be, and I mean, not all the compounds are going to be present all the time, because the time of harvest is important, the season of harvest is important, even the stage of the harvest is important. Okay, so I am not going to put so many things, like we have done research, photosynthesis improved, I mean, nutrient content improved, nitrogen content, especially the whole lot of transcriptome work we have established, how without having any significant nitrogen compound, why it is important, why the nitrogen content is increasing. That is because there are two genes that are regulated, upregulated, that is nitrate reductase and the nitrogen transporters present in the roots. So that is what we have done, and we have published, I think, more than 50 research papers, especially targeting each of the mechanism, each of the scientific mechanism. I can share the presentation with Vipinji, so you can have a look at all the publication of whatever is your interest. So I'm not going to go into the detail, and that's a very good, I mean, disease resistance mechanism, how it is important for soil microbes. This is, for example, soil microbes, how it is important. Even under drought condition, it is going to increase the diversity and population of the soil microbes. This we have done through metagenomics, 16S rRNA, next genome sequencing. And this is also good for, I mean, from policy point of view, this is my last slide. Policy point of view also, it is important because This is a extremely low carbon footprint product. We have calculated about, I think, 118.6 kg carbon dioxide equivalent per kiloliter of the production of biostimulant at industry level. This is the industry data, and we have published in several of the research papers. So ultimately, when you use low carbon footprint product by reducing at least 25% of the recommended dose of inorganic fertilizers, the carbon footprint per unit of production is much more reduced. So this, thank you. I mean, that is all I think for today. Thank you very much.